Welcome and hello everyone. Today our group will present our project entitled Milk Coagulation using Rennet Enzyme. Milk coagulation is an important process in the dairy industry, especially in cheese production. Liquid milk is unstable for long-term storage, so proteolytic enzymes such as rennet are used to trigger curd formation. Rennet acts as a catalyst by helping milk proteins form a solid structure called curd. This enzymatic process is widely used in commercial cheese manufacturing to separate solid curd from liquid whey. In this experiment, we investigate the effect of rennet enzyme on milk coagulation and curd formation.
Milk is mostly water, but it also carries proteins called casein and minerals like calcium. Naturally, these proteins hate water and want to clump together. However, nature has a clever way to keeping them suspended so milk stays liquid. The proteins group themselves into tiny protective balls called micelles, which you can see in the diagram on the left. Inside these balls, we have alpha and beta caseins. They hate water, so they hide deep inside the center. On the outside of the ball, we have K-casein. Think of K-casein as a layer of tiny hairy shells that love water because these outer hairs carry a strong negative electric charge. The protein balls act like magnets with matching poles. When they get close to one another, they repel and bounce away. This bouncing is what keeping the milk perfectly mixed, smooth, and liquid. To make the proteins stick together, we add an enzyme called chymosin. You think of a chymosin as a highly precise pair of microscopic scissors. Instead of digesting everything, chymosin goes after one exact spot on the protective hairy casein outer layer. It snips the protein chain right in half. We call this a molecular haircut. As shown as in the chemical equation at the bottom right of this slide, the snip splits the shield into two parts. First, we get the para-casein. This is the water-hating part that stays attached to the core of the protein ball. Second, we get the glycomicropeptide. This is the water-loving negatively charged hair that get cut off and simply drift away into the liquid way. By cutting off these protective negatively charged hairs, these protein balls lose their ability to repel each other. They can no longer bounce away. Their sticky water-hating cores are now exposed, allowing them to crash into each other, stick together, and form the solid curd network we are looking for.
The activity of Rennet enzyme is influenced by several factors such as temperature, enzyme concentration, and pH. The enzyme works best at an optimum temperature. If the temperature is too high, the enzyme may become denatured and lose its activity. In the dairy industry, Rennet enzyme is widely used because it improves coagulation, efficiency, and helps produce cheese with better texture and quality.
Next, for the two-phase coagulation model. First, primary enzymatic phase. The chymosin cleaves K-casein at the free med bond, releasing the soluble glycopeptide into the milk serum. Then, the secondary aggregation hydrophobic para-casein micelles are exposed. In the presence of the calcium ion, micelles start to clumping. So, they just start to creation. Clumping micelles form a three-dimensional network structure trapping moisture and fat globule inside. So the synalysis, which is separation, the matrix contrasts actively squeezing out the watery whey and compacting into the solid cheese curd. For the experiment design and planning, the scientific hypothesis, milk treated with rennet and milk treated with vinegar will both coagulated, but they will produce the different curd and the characteristic. For the experiment variable, the independent is coagulation agent used, which is rennet and the vinegar. The dependent variable, the formation of curd, coagulation time taken, and the controllant variable is the milk volume and the temperature of the milk. For the experimental metric, the test tube A is the treatment made with the rennet and the test tube B treatment with the milk and the vinegar. They both using the same temperature make, which is 40 degrees Celsius. And the test tube A biologically purpose to investigate how the enzyme chymosin in the rennet coagulated milk protein casein through the enzymatic action mimic the process used in the cheese production. And the test tube A expected coagulation produce a looser crumpled curd that break apart more easily than the vinegar treat sample. And for the test tube B, biological purpose is to investigate how the acidify lower milk pH and cause casein protein to precipitate, resulting in acid indulgence coagulation. And the test tube B expected coagulation is to purpose a film and cohesive curd.
Next, I will explain the lab protocol used in this experiment. We begin by preparing 2 samples and adding fresh milk to each beaker. The milk was heated to the required temperature and then removed from the heat source. Rennet was added to one sample, while vinegar was added to the other, and both mixtures were stirred gently. The time taken coagulation was recorded, after which the mixture was filtered to separate the curds from the liquid. Finally, the texture and appearance of the curds were observed and compared.
For the result, the test tube A, which is made with the rennet, coagulation time it take 40 minutes. Observation rapid formation of a softer, more crumpled curd due to the acid coagulation. And for the test tube B, made with the vinegar, it take the 42 minutes. The observation gradually formation of a film, smooth and the elastic curd with whey separation.
Based on our observation, the milk treated with rennet enzyme coagulate faster and form more curd compared to the control sample. This happened because chymosin in rennet successfully broke down casein and caused the milk protein to coagulate. So, from the experiment, we can conclude that rennet enzyme plays an important role in milk coagulation and dairy processing. The enzyme improves curd formation and is highly useful in cheese production. Overall, this experiment helped us understand the function and application of enzyme in the food industry. That's all from us. Thank you.